Thank you. Oh, well, let's talk about biochar again here, but this time in relation with its effect on the long-term effect, rather, on soil enzyme activities. Now, a little bit about soil enzymes and a bit about soil enzymes. Soil enzymes are involved in nutrient cycling within the soil. They play just a tremendously big, important role in soils as far as mineralization of organically bound nutrients such as phosphorus, sulfur, and nitrogen. And I have to pretty much look at the screen because I can't see everything in here, but I pretty much know what's going on here. At this stage, I'd better. Enzymes are catalysts, they're proteins, as we all know, and they participate in chemical reactions, bring the activation energy down. They are specific in their operations, they know what they're doing, and they attack what they want to attack. Certain bonds desirable to certain enzymes, and certain other bonds they don't care much about. So they know what they're doing in the soil environment. So enzymes are denatured by elevated temperature, and obviously one thing that's important here, they are also denatured and deactivated and inhibited by ionic strength. So pay attention to that ionic strength, you know, right here. And pH, of course, plays a role because they have an optimal pH that they just love to do their work. Okay, they're picky. Proteins are picky. And then talking about again about the knowledge about the biochar that's like right now is increasing exponentially, and everybody is doing everything, which is good. Knowledge about the effect of biochar on soil enzyme activities is scanty. And there are actually contradictory statements in the, or findings or declarations and assessments and assertions in the literature. Some, they say biochar blocked the binding sites of enzymes and deactivated the enzymes. And some say, oh my gosh, biochar did a miraculous thing to the enzyme and made it work and all that. And you wonder and scratch your head and say, why? What's going on here? And well, I think I know just a little bit what's going on. And I'm here to share my little knowledge with you. And I appreciate you being here. Objectives of this study is how does the presence of biochar in soils or in a soil affect the activities of soil enzymes? Picked up a Kent, you're the lots of enzymes, all right? We picked up acid and alkaline phosphatase, they call them monophosphomonoesterases, and arylamidase and beta-glucosidase, and then how do biochar produce from different precursors of ligneous herbaceous biomass affect the activities of these enzymes? Does the rate of applied biochar affect enzyme activities? See? All right, now, this is what phosphomonoesterases do in the soil environment. What they do, as I said, they go after these peculiar bonds of phosphorus, and they hydrolyze those bonds, and they release the phosphorus, and the plants are capable of absorbing that available phosphate or phosphorus. That's what they do. And these statements come mostly from M.A. Tabatabai, who served as my advisor a long time ago. And arylamidase is involved in nitrogen mineralization within the soil. So if it has higher activity in the soil, that means that the soil is producing mineralized nitrogen available to plants. And beta glucoside is actually with carbon, and microorganisms need energy, and they use that to get some of the energy they need. And the biochemistry of it is available everywhere. You can refer to that. In this study, this is the soil, the same soil that I mentioned this morning for those of you who missed it. This is the soil, cultivated alpha soil, and this is pretty much routine analysis of some of the chemical properties of that soil. And then this is our treatments. Say no biochar, and then soybean and biochar, and 2% miscanthus, 5% miscanthus, 2% and 5% pine biochar. Miscanthus and pine biochar, yes. All right, in greenhouse experiment, 1 day, 3 day, 10 day, 30 day, and 60 days, we sampled the soil from the pots, 5 kilograms each. And we had applied phosphorus to our soil, and some additional data that I mix in later on, just give you a warning. And this is basically what the technique that we use by IVS antibody for acid phosphatase. Yeah, pay attention to right here. It says weigh 1 gram of soil. You know, there are some studies that what they do, they just weigh 1 gram of biochar, and then they add this whole thing, and they say, well, biochar inhibited enzyme activity. It's like within the soil we have montmorillonite smectite. And if you go and weigh 1 gram of smectite, and you add to run this test, you realize that smectite really inhibits enzyme activity. But at 2% rate, there is little information. Or at 5% rate of biochar to the soil, how would that function? How would that rate of biochar affect enzyme activity? That's the real world. Let's talk about the real world. You know, and this again, let's pay attention that 1 gram. Of course, I promised you that they can add 1 gram of biochar and run this test, because then they have to modify the technique, the method here. They probably weigh about 0.1 gram, but that doesn't matter much. And the same thing, 1 gram again, huh? But how did we produce our biochar? This is important. I've already talked about it, so I'm going to just touch upon it and go through, because they're going to stop and they're going to say, you've been talking way too much. So here's biochar. This goes, to be honest with you, the biochar industry may not even survive without that component, which is called bioenergy, biofuel, okay? That has to be part of this scheme. Otherwise, this on its own, you can use it to your pot in your house and all that, but on a large scale, forget about it. And, okay, this is how I produce my biochar. It's inner barrel, outer barrel. The biomass is in the inner barrel, and I burn the biomass in the outer, between the inner and outer, bring it up to overcome the activation energy, and then the system becomes sustainable and burns on its own and produces lots of good biochar. It's a mobile unit, it rains, bring it and put it someplace else. All right, now temperature 500 degrees, we have reached a temperature of 617 degrees using this homemade pyrolyzer. That's the biochar from miscanthus right there. Then we crush the biochar for the woody biochar, pass through 1 centimeter screen, and then use it that way. You see, you can use powder on a large scale or even small scale operation. It's just not practical. The dust is not good for you. I don't know if you have worked in any coal mine. You don't want any of that dust in your lungs. At any rate, then mix it with the soil and talk about the results here. Results and discussion here. And here is the pH of biochar I mentioned this morning. You expect biochar to have different pH. Biochars are different, all right? no, you can't just say biochar. You could say biochars or biochar types. You know, many factors involved there. The biomass precursor, how you produced your biochar, this and that. Okay, nitrogen. You see the herbaceous biochar, lots of almost nitrogen fertilizer there. Okay, here's what one of the things is that. This is put all our data together, okay, and say, okay, how do the, I'm going to look at the effect of biochar and enzyme activity in the soil, okay? Day one, go to the, you see, look at those acid phosphatase there, okay? This phosphatase, acid phosphatase, likes lower pH. It's not really low, just like about around 5.5, 6, okay? Okay, here's how it is. It goes right up. It just goes statistically the same, a little bit different. Different at 5% is different, is another one, different, all right? So in other words, it goes up. The activity goes up with time, okay? With biochar, there, it doesn't matter what kind of biochar, just some or lump them, everything together. Look at this one, the alkaline phosphatase. You see, right? At the beginning, low. So you add biochar, the pH goes up, the alkaline phosphatase likes it, they're all up. Good. So what is this deal about biochar inhibiting enzyme activity? But wait. Okay, look at acid phosphatase, alkaline phosphatase. Well, acid phosphatase is predominant in soil. It is really the bulk of phosphatase activity is acid phosphatase. Okay, so no matter what you do, you have acid phosphatase, more activity for acid phosphatase. Okay, within the pH that we are dealing with. Well, look at this one, allyl amidase. Okay. It's at BC, okay? It goes to A's. This is different from this. Then it's low, significantly low. Then it goes up, and then back up to this level. If I were to stop my experiment after 10 days, I would have concluded that biochar reduces enzyme activity. I mean, this enzyme activity, allyl amidase. And I would have been wrong. Unless I would have, if I had said after 10 days, at 10 days it reduced it, probably I would have been right, but somebody would have asked it, what if you had waited another week? Okay? Now, what is this one? Beta-glucosidase, pretty much the same thing. High, going down, going back up. You see what it means? Long-term effect of biochar on beta-glucosidase. You see that it recovers, the system recovers. You don't tell me to interpret this data. Okay? Don't ask me that question now because I don't have much time. The type of biochar effect on acid phosphatase. Now look, the types of biochar. You see, right here you see that not only the type of biochar, but the rate of the application of biochar can affect the processes within the soil environment. Physical, chemical, physiochemical, biochemical, whatever, all right? No biochar. Look at this one. Look at this one. Then look at this one here. This miscanthus biochar, it's lowering it. And when you go to 5% miscanthus, it's lowering significantly back down. So what happened? The rate of biochar affects the enzyme activity, as if the biochar brings ions and dumps it in the soil, and that ionic strength affects the enzyme activity. Just one interpretation of that. There are several more probably. Types of biochar, you know, see, soils are very complicated systems, just like a human body almost. Types of biochar, effects on alkaline phosphatase. Now look at this one. Pine biochar is bringing alkaline phosphatase down. Or is it really pine? Yeah, probably. Is it because pine biochar has a lower pH value? Or is it because there is some trace element or micronutrient there that has inhibitory effect on this enzyme's alkaline phosphatase activity. Okay, the pHs. This morning I showed this one. Look, the same almost pine biochar doesn't do much to pH. But look at the herbaceous biochar. Increases the pH significantly almost. There. Okay. Aryl amidase. Aryl amidase is peculiar here. Remember that has something to do with nitrogen cycling and all that. So look at it this way. Okay, look at the pine biochar. And then you add 2% or 5% of miscanthus biochar, aryl amidase activities go up. Well, you would say that you increase the pH, the optimal pH for aryl amidase activities 8.5. Perhaps you helped it that way. Maybe. Yeah. Look at this one. Beta-glucosidase, no biochar. And look what happens here. Well, actually, beta-glucosidase is happy with no biochar. Look, you come here to 5% biochar, decrease. 2% miscanthus, decrease. There's a 5% that says inhibiting it. Look at that. And that's beta-glucosidase. Why? How? You see, these biochars have different effects on different enzymes. You see, enzymes are very sensitive. Some people have used them as, or even talked about using them as an index for soil quality because they're sensitive. You add something to that system, the soil system, the enzymes immediately react to that factor or that outside intrusion. See? All right, conclusions here. Effect of time. The overall activities of enzymes studied in this greenhouse experiment generally increased throughout the course of this study, which is like 60 days, and stage R6 for soybeans. We cut them off, we stopped the experiment. You see, we could have gone longer. Also, when you use biochar, mix your biochar with 6 to 8 inches of your soil. Just don't broadcast your biochar on your soil. Otherwise, you won't see much of any difference, all right? You may see a little bit of difference increasing in yield and all that, but you lose most of it to erosion, wind, and water. You have to incorporate it into your soil, mix it with your soil. Effect of biochar type and rates. Biochar type and in its application rate affected the activity of acid phosphatase. As 5% miscanthus biochar lowered the activity of this enzyme significantly, I think because of ionic strength there. At 2% application, I noticed that you stood up, that means I have only 2 minutes. All right, okay. At 2% application rate, however, this effect was not significant. You can read most of that. Okay, the pine biochar reduced alkaline phosphatase activity. That's the activity of aryl amidase increased significantly by the presence of miscanthus biochar, regardless of the application rate. The pine biochar, on the other hand, did not affect the activities of this enzyme. And the activity of beta-glucosidase was significantly lowered by miscanthus biochar. The inhibition was exacerbated at a higher rate of application, but the pine biochar did not affect the activity of this enzyme. I'm thinking that might be my last, because I timed this one just right. So you can, yeah. So, no, I went to Russia last year, invited me to Russia last year, and they were trying to rebuild their country and all that. And I walked into there, and so they move around and all that. And I said that right there, as soon as I got off the plane, I said, wow, everybody's Russian. And my friend said, of course they are Russian. But actually, I mean the other Russian. They are Russian. And you were Russian, you know. Thank you. I will let you do it because I don't want to mess it up.
Well, thank you very much.
Thank you.